Let's talk about ProtVista, a component which is heavily used throughout the PDBe-KB aggregated views. In this, ProtVista is essentially a sequence feature viewer, which was jointly developed by UniProtKB, Interpro at EMBL-EBI. And this is freely available for anyone to use at GitHub at this link. Now, here you can see in this viewer, this is the protein sequence, a scale from 1 to the total length of the protein. You have ProtVista main component or main section and the subsection, which shows you annotations for various residues. You can collapse by clicking the arrow or the main ProtVista section. If you hover over any region or click on its details, you can see the details of that particular residue on the tooltip. There are icons like this circle where you can focus on a particular subtrack, and you can hide a particular subtrack using this cross button. In ProtVista, you will see evidence-based section in blue color, and you will see the prediction-based section in gray color. You have a reset button, which resets the zooming and the hidden tracks and takes you to the default view. You have a section for navigating the range where you can put or highlight a specific region of your interest. If you highlight it to high level, you can even see the amino acids. You can use your mouse scroll as well in order to zoom. You have a special button for hiding the various sections. For example, here I have hidden PDB structures and secondary structures. So now let's talk about the structures section information in aggregated view of proteins. This is how the structure page looks like for a given protein. Here we are looking at spike glycoprotein. It mainly has two sections. First, you have the 3D viewer showing the PDB structure of the protein. And then you have the ProtVista sequence viewer, which shows you all the structures and various other information. You can see by default, the structure which is first listed in the ProtVista is highlighted in the MolStar viewer. And if you want to view the specific PDB entry page, you can click on this button. In order to see, you can click on any segment which you want to see in the MolStar viewer over here, and the structure will be displayed. For example, over here, if I click at this section, it highlights 7SO9. Then in this section, you see at the top, experimentally solved structures which we have in PDB. Then we also have data for predicted models from various resources like Swiss model, SASDB, and also AlphaFold-DB. This section gives you an idea of the region for which we have the known structure. In general, the PDB entries are sorted by maximum number of observed residues and resolution. If you look at the details for each of the subtrack in PDB structures, you will see there are links and the button is highlighted as green if it is involved in a complex or if it has interactions with DNA and RNA. In this case, there is no structure which is bound to DNA and RNA, hence it is grayed out. You can also see the link to various structures to which any ligand is bound. Towards the end, you see the resolution of that structure. In case of polyproteins, you also have processed proteins, and you can see the location of various processed proteins, where they are chopped off in the polyprotein. And this view enables a quick identification and comparison of various processed protein structures. You can also see the secondary structure elements which are observed for a representative PDB chain for each of your covered region. You can also see domain information. We have domain information from PFAM, CAT, SCOP, and even Interpro, which is highlighted in this section. In terms of flexibility, we have data coming from MobiDB, which basically gives a consensus disorder prediction from UniProt sequence for a given UniProt sequence. For more of the details about our other partners, we will talk about it in annotation section. You can view the 3D view of superposed structures as discussed in the summary section from this place as well. It will open a 3D viewer showing you all the superposed structure for any given region. You can download the data by clicking on this button for all the structures. For example, in this case, I can download all the 890 PDB entries corresponding to this particular UniProt accession by the click of single button. And I can choose MMZIP file, FASTA files, or even validation data. All the annotations which you see in Prot Vista Viewer can also be downloaded in CSV format by the use of annotation button.